Let's learn the order of draw Milka's way. Now, I remember when I was learning to break down the order of draw, I did it in a form of flashcards, which is one easy way to learn them. I broke them down into different categories, which is our blood cultures, coagulation studies, clotting activators, heparin, EDTA, sodium fluoride, and different types of anticoagulants. Now, remember the purpose of the order of draw. It's to prevent cross-contamination. Know that certain additives can mix with other additives in the correct order of draw. Therefore, sodium citrate can mix well with heparin, but if we were to do the opposite, we can see different traces in the sample when it's down at the lab. Now, let's start off with our blood cultures. Blood cultures are the first sets of bottles or samples that we would collect when it comes to the order of draw. Even with the blood cultures, it has its own particular order of draw, and that is from aerobic to anaerobic, from oxygen to no oxygen. When your doctor will order a blood culture, it's to check if there is any bacteria or infection in the blood. We will come over, we'll come across terms like sepsis, and that's poisoning in the blood. The different type of antiseptic that you can use is ChloraPrep chlorhexidine. You will use a butterfly to collect the sample. You can use the syringe method, or you can just uh, apply the bottles to the end of the butterfly needle. They come with their own hub. Our next set is our coagulation study. Coagulation studies is our light blue tube. Our light blue tube has an additive of sodium citrate. It is an anticoagulant. So therefore, if we were to spin it down, we would receive our plasma. The light blue tube is the only tube in the order of draw that you have to invert, properly mix the additive with the blood at least 3 to 4 times. Now be careful because this particular tube has to be filled to the fill line. If not, the lab will say to you that you have a QNS, which means quantity not sufficient for the amount of blood that you sent. They cannot perform that test. TNP, test not performed. The test that can be performed with a full blood sample from the light blue tube will be our PT, which is our prothrombin, PTT, PTINR, and D-dimer. These are important tests when it comes to our coagulation studies. These tests will check how long it takes for your blood to clot, what are your platelet counts looking like. All of that information can come from our coagulation study tube, which is the light blue. Now let's talk about our clotting activators. Our clotting activators, we have our red top, tiger, and our gold. The red top, remember that it's different from our SSTs, which is our serum separating tubes, because it does not have the gel barrier at the bottom. But our SSTs, like our tiger and our gold, does have that gel at the bottom, which is called polymer gel. The difference is when you were to spin down your red top, although it will give you serum, but there is no gel to separate the serum from your red blood cells. So the way that you will transfer the serum to another pour-off tube is by pipetting the serum. Be careful because you may have a little bit of red blood cells going on there, depending on how you pipette the serum out of that tube and to transfer it into a pour off tube. With an SST tube, because that gel barrier is there is no need to worry about the serum mixing with the red blood cell. With your red top, this has no additives. It's considered to be a non-additive tube and a discarded tube. That means, well, I like to call it my boo-boo tube. If I make a mistake, I'm able to clear the line using my red top and then continue with the correct order of draw. And discard it, throw it out. They both give you serum because they contain clotting activators inside of those tubes. Now, with a serum separating tube, that's your Tiger in Gold, it needs to sit to clot for at least 30 minutes. If you were to spin it down before that, you may risk your sample becoming hemolyzed, and that is the destruction of red blood cell. And the way that our serum will look, it's like a reddish, um, pinkish color, a tinged color. Okay, that's an indication that our sample has been hemolyzed. When we look at our sample of an SST and it is now broken down because it's been centrifuged, you will see the serum at the top, and then you have your buffer coat in the middle that is containing your white blood cells and platelets. And then you'll look underneath that, you will see your red blood cells. This particular tube will be sent to chemistry. Okay. Now, our next set is our heparin. Our heparin comes from a green tube. It has sodium heparin or lithium heparin. These two tubes are considered to be an anticoagulant tube. Again, it's collected whole blood and it cannot clot. An important test that goes along with our heparins, our green, is ammonia, arterial blood gas, and these two tests need to be chilled on ice. Don't forget, these are considered to be a special handling test.